and talk about phenotyping and talk about a particular type of phenotyping, which is probably one of the oldest types of phenotyping, which is histopathology. And this really is a reflection of some discussions that we've been having in the IMPC for about the last 4 years, something like that, which has actually led to some very careful considerations and interesting conclusions about the use of histopathology, traditional histopathology, in mouse phenotyping, what it can contribute, and also its place in a primary phenotyping pipeline. So I want to start really talking about the high throughput histopathology as part of primary phenotyping. The first question is, well, what additional information can it provide? So you're actually now all very familiar with the primary phenotyping pipeline of IMPC and NeuMoDIC. People have been talking about that for the last few days. Particularly, what can it contribute to the IMPC efforts? And we were also discussing things like technical feasibility and logistical feasibility, whether it was cost effective. And so a lot of these considerations came up. We had a meeting in France in 2010 where we actually thrashed out a lot of these issues. And so what I'm going to do is to present you some of the conclusions from that meeting with some examples, particularly of how histopathology can be very useful in finding phenotypes that you can't find other ways. There's a couple of publications which really contain most of the points I want to make, which again you can go away and look up. The meeting I referred to was actually published, there was proceedings published in Toxicological Pathology in 2011. And then a lot of the points were raised in this commentary in Disease Models and Mechanisms, which came out last year. So a lot of what I'm going to say is in here, but some of the more specific examples I want to show you obviously aren't. So what can histopathology contribute? Well, the first thing is it gives you both deeper and broader phenotypic information. You're now all totally familiar with the concept of phenotype ontologies and information content and how important it is to actually find phenotypes which are discriminating between different diseases. So a phenotype like hyperglycemia or mental retardation is a very undiscriminating phenotype because so many human diseases have it as part of their makeup. So these have low information content. And what we really need is annotation with a higher information content in order to discriminate between different human diseases. It gives us contextualization of other findings, which, as you'll see, is very important, and often an etiology of the overall disease processes that you're measuring and going on with other methods. Of course, it covers nearly all organ systems. So nose to tail histopathology covers all organ systems. And even as a primary phenotyping, one could actually take around 45 or 50 tissues at high throughput. And that actually covers an awful lot of the animal. Detection of subclinical disease, as you'll see, is actually very important. So these are diseases which are actually incipient but aren't manifest. And again, this is particularly important with age-related diseases. Bear in mind, actually, as I'm talking, that the primary pipeline of the IMPC ends at 17 weeks. So there's no phenotypes collected after 17 weeks. And that's actually an issue in terms of getting hold of age-related diseases. And then there are phenotypes which are just not detected by in vivo assays. So unknown unknowns, I hate to say this, but it's actually really good, unknown unknowns. So things you don't know, things that are completely unexpected. And then there are known unknowns, where you don't have any pups, you know there's something going wrong, but you don't know what it is. So you know there's something wrong, but you don't know what it is. And then you have this group of mice with no phenotype, or very poorly defined phenotypes. And actually probably now more than 10% of mice with no phenotypes actually have a phenotype by histopathology. So this is a very salutary lesson, and of course it's not the first time that anybody's mentioned this. It's about context. So this is a very important poem, professionally for all of you here, it's a very important poem. It was 6 men of Hindustan, to learning much inclined, who went to see the elephant, though all of them were blind, that each by observation might satisfy his mind. And so these men of Hindustan disputed loud and long, each in his own opinion exceeding stiff and strong, though each was partly in the right, but all were in the wrong. And this is what happens when you actually use a multiplicity of assays to assay the same organism, but from different perspectives, because each assay doesn't see the results of the other assays. And so, here we have, somebody having a look at the tusks here and saying, well, it's a spear, obviously. And somebody looking at the ears and saying, well, no, it's obviously a fan. And so they're all right. Yeah, it looks very much like a fan and it looks very much like a spear, but actually they're looking at an elephant. And so you have to contextualize your findings. And that's really the key thing. There's a couple of little examples here from the Schofield aging program. So mice which are pulled out as phenotypically abnormal with a gait abnormality. They were doing a gait assay, with little footprints and looking at gait. And they thought, well, maybe these mice have vestibular lesions or cerebellar lesions because they're walking like this. Well, actually, what they had was cancer. And they had metastases to the lymph nodes, and they had lymphomas. And the lymphomas were sufficiently large to stop them walking properly. So the gait abnormality is a cancer phenotype. You don't know that until you've actually cut the lymph nodes and seen that they're full of lymphoma. An example I was talking to Glauco about earlier, very high erythrocyte counts, which were coming up on and off within a cohort. And those were discarded as noise because it was not possible for a mouse to have this many erythrocytes, so it must be a machine error. No, it was erythroleukemia. It was the phenotype. So actually, being able to interpret the assays that you're carrying out is very important because that interpretation might be very unexpected. And the histopathology gives you that kind of contextualization. So the pathology integrates the data from all these multiple sources and actually gives you the whole phenotype. And you have a much higher chance of identifying cognate human disease. So here's a guy saying, well, it's a metabolic mutant, or it's a vestibular mutant, or a back to front mutant, or whatever, and of course, all right and all wrong. So let's look at some instances of detection of subclinical disease. This is obviously particularly important for age-related disease, because some diseases that might develop later on in life do have subclinical manifestations, but they may not actually be evident at the time when the assays are done. And a lot of robust phenotypes, really robust phenotypes, fully penetrant phenotypes, are not visible by 15 weeks. And some of these are important phenotypes like cancer and degenerative lesions, and they're just not detectable by physiological or behavioral assays. So here's a knockout of a gene, ectonucleoside triphosphate diphosphohydrolase. This is work of Peter Vogel. At 44 to 70 weeks showed frank hepatocellular adenoma and hepatocellular carcinoma. And actually, although there wasn't much of an effect in the first sort of 20 weeks of life, they begin to lose sperm and enter full spermatogenic arrest later on in life. But at 14 weeks, they're showing foci cellular change in the liver. So there's no measurable alterations by any assay other than histopathology. But here are these foci cellular changes. And here are degenerating hepatocytes from these young knockout mice with these hyperinsulinophilic shrunken hepatocytes. And so you can actually tell this is a phenotype, but it's a phenotype which is going to be a very important phenotype later on in the life of that mouse. And it is characteristic of that particular lesion. And yet, if you just simply do, if you don't do histopathology at these young ages, you never see it. This is another one. This is the ATG4, the autophagy-related 4 homologue gene. This was tested extensively between 12 and 16 weeks. So all of these tests came out negative. Locomotor activity, sensory motor gating, etc., learning and memory, open field, tail suspension, etc. I don't like to mention the rotor rod assay again, but there was a trace of activity in the rotor rod assay. So maybe rotor rod's not so bad after all. I had to say, everybody said something about rotor rod, I think. But actually, if you look early in life, what you see are these lesions in the cerebellum. So you see these protein accumulations, which you can actually just see here are the featureless protein accumulations. You can see them here later on. This is the similar time point actually stained with for ubiquitin. And so these protein aggregates are accumulating ubiquitin. And if you actually have a look at where they are, these are actually manually marked up for the lesions, so you can actually see the overall distribution. You get these amorphous globular bodies at 12 to 14 weeks in the neuropil of the deep cerebellar nuclei. And so this goes on to have profound cerebellar lesions, and yet, again, there's no way of picking this up in the mice at the stage using behavioral assays, at those early stages using behavioral assays. This is actually just a little example I put in because there's one or two interesting points here. So we're going back to PhenomeNet a little bit here. So this is the knockout of the Enzyme 2 gene, which was carried out at the Sanger as part of the mouse genetics program. And this is a picture of the phenotypic profile from the Sanger database. And these Enzyme 2 knockouts showed quite an interesting set of rather varied phenotypes. But actually, on closer inspection, he says optimistically, turns out they have a skin phenotype too. But the skin phenotype wasn't immediately apparent, particularly not in the mice at this early age, and they have an abnormal exogen phase. So what's really happening here is that as the hair cycle progresses, the old hair is lost and the new hair comes through. Then the old hair, the club hair, actually is not shed properly, so it's actually retained. So there's a very marked abnormal exogen, and here's a nice, quite rare picture down the whole of the length of the follicle. So these mice have abnormal exogen, and so this was another phenotype that wasn't detected with the standard primary assays. We actually took all of those phenotypes, including the skin phenotype, and put it through PhenomeNet. You can see that was an old PhenomeNet, and we came out with this gene MRT5, which slightly surprised us. But actually, looking at the phenotypes of the MRT5, locus were actually very reminiscent of the Enzyme 2 mutants. And then this was actually on 5p15, but not actually pinned down to an individual gene. And then shortly after we actually brought Enzyme 2 up as a potential candidate, a paper was published, an abstract of a paper was published at the American Society of Human Genetics, not last year, but the year before, identifying Enzyme 2 as this pathogenic gene for MRT5. So we'd actually successfully predicted from that group of phenotypes what the gene was going to be. And Enzyme 2 has also been highlighted by the Watt lab actually as well as an important gene in hair biology. So this is an interesting story. There are also advantages of doing your histopathology systematically. This is another example. So this is the Porcupine homologue gene. It encodes an endoplasmic reticulum protein, which is involved in the processing of Wnt proteins. There, I think, still is only one knockout available, which you can find in MGI, which shows this focal dermal hyperplasia and skeletal defects. But there's not much else in terms of the description of the phenotype there. Looking at the skin, as part of a systematic survey, then you find these focal areas of dermal hyperplasia. So you can actually see this area here, and next to it there's this area here, which is an area of perfectly normal formed dermal collagen. And this is your blow-up of the area of focal dermal hyperplasia. So it's a very striking aspect of the phenotype, which was never reported in the original mouse, although it's almost certainly there, in fact. But people just didn't look at the skin. They didn't do the pathology. Not only did they not look at the skin very carefully, they didn't look at the teeth either. And so this is a normal tooth bud, and this is the Porcupine knockout. And you can see that all these irregularities and discontinuities in the odontogenic epithelium. You see there's a gap there, there's a folding here, there's what's probably a kind of folding going on here. So the odontogenic epithelium is severely disrupted in this tooth. You can see similar disruptions here and here, and some of the teeth are just completely non-viable. So there's a very strong dental phenotype here, which hasn't been picked up in the phenotyping previously. And if you look at the description of the human disease for the same gene, in fact, you find that the teeth phenotype is very, very strong and almost exactly the same. It has hypodontia, dental dysplasia, enamel hyperplasia, malocclusion, etc. So actually, the mouse and the human match really very, very well indeed in that particular syndrome. But people didn't look at the teeth. So what the message from this is that there's a huge amount of advantage to do your histopathology systematically, because you will pick up things which you didn't expect. For age-related diseases, of course, one of the issues, if you look at the major forms of human mortality, then cancer and cardiovascular disease are the two major sources of human mortality, and they are age-related diseases. So in terms of usefulness against major human diseases, age-related diseases, diseases which have onset with age, are actually very important. As I pointed out, you actually often can't see good phenotypes before 15 weeks. So this phenotype, this muscular dystrophy phenotype, was seen at 12 weeks. And actually, even though it looks quite severe histologically, there's no behavioral or locomotor issues were picked up. And in fact, actually, even if you look at MDX mice are not particularly weaker than the wild types. So there's something about dystrophic muscle in mice, the pattern of muscular dystrophy, which doesn't produce the same kind of weakness as it does in humans. So you can see it histologically, you can't detect it with an in vivo assay. The same with this example of neurogenic atrophy. Again, the only way to pick this up is actually histologically. It couldn't be picked up physiologically. Here's a nice, this is a frizzled 4 mutant. At 12 weeks, 20 weeks, this is the wild type. And at 12 weeks, the wild type, the homozygote doesn't look really remarkably different from the wild type. But by 20 weeks, you have marked degeneration of the retina and the beginning of this this exudative vitreous condition. And this was not picked up with eye assays either when the eye assays were done. So I think those are just some nice examples to show you that doing a complete histopathological phenotyping can give you an awful lot of stuff back that you missed. And it can also give you explanations for stuff which is coming out of your physiological in vivo assays. And one of the issues for the pipeline, and this may be of relevance to some of you or people who are sort of setting up phenotyping, is exactly how much material can a pathologist handle, can be expected to handle, and what kind of cost are you looking at. And the consensus of the meeting was that basically 2 pathologists should be able to handle 20 lines a week, doing 2 mice of each sex for each line. So two pathologists, you actually need people to process the tissue, which is actually the most manpower intensive thing. But in fact, it should run on about $1,500 or less per line, excluding the salaries of the pathologists. So actually, it's not incredibly expensive. One of the things which also became apparent was the issue of who actually looks at the sections in the primary screen. And actually, whether the pathologist picks up lesions in the primary screen really depends on how expert they are. And although there were some suggestions that someone less trained, or what people kind of call a parapathologist, might be kind of useful at the coalface doing the large-scale primary screening, in fact, they would almost certainly miss things. And you want your most expert pathologist to see the things straight away. So there's actually quite a nice example here of retinal degeneration. So here are two retinas. Here's the wild type, here's the homozygote. The difference between these, the actual mutant phenotype here, is actually in the relative thicknesses of the granular layer and the cell body, the cell bodies. And in here, this is the outer granular layer is very much decreased in thickness. You need somebody who is really very experienced to spot that on a high throughput screen, for example. Having somebody who is an expert, fiddling under the bonnet here, looking at the material as it's generated, I think is actually very important. The expertise base for mass pathology has been a cause of some concern. So the number of people who are used to looking at mice, never mind laboratory mice, is actually relatively small. And so there's been quite a bit of literature and discussion about the training of mouse pathologists. In fact, the situation is not quite as bad as it was a few years ago, in that quite a few young pathologists have come through training and decided that this kind of research work, looking at mutant mice and building models of human diseases, is actually quite attractive. And so, in fact, there's quite a few people coming into the mutant mouse world, as it were. There are some new initiatives, particularly in France. And so the French have been very proactive about training mouse pathologists. There has been less proactivity in the UK, for example. I don't know whether there's been anything in Italy. But there are some courses. There's a course run at the Jackson Laboratory, and there's also an online course run by the University of California at Davis, which, although will not actually give you full training in mouse histopathology, actually are extremely useful and good introductions to the mouse for people trained in human or veterinary pathology. So there are courses available, and people are very generous with their time. So this was the other idea, that actually a network of distributed experts, because now it's very easy to share images, and particularly, I don't know whether any of you have seen this kind of zoomable histopathology images, where it's as if you have the microscope, you have the whole slide, and you can zoom in and zoom out and move it around. These are now such high quality that image sharing sort of internationally is very easy. And a lot of people increasingly are doing that to get second opinions, to get help on particular systems. It's hoped that some of the mouse clinics who take up the pathology might actually act as training centres. So histopathology, having done all these discussions, has been accepted into the pipeline as an optional assay. The only IMPC centre which has taken it on as routine is the Toronto Centre for Phenogenomics in Canada. They've actually set up their own primary pathology pipeline, which will really be the model for the others if they actually set the pipelines up. And that's very interesting. So here's the TCP. Nice, building is still nice, new and shiny at the moment. They actually are capturing pathology data using ontologies. So we're coming, I'm afraid, back all the way to informatics again. They're capturing it using ontologies for all of the reasons that I mentioned earlier, that we need a way to capture data which is unambiguous, which can be used computationally, which can be used to search and categorize data and phenotypes. So they have a database into which they put both gross pathology and histopathology data. I'll just show you a couple of shots from that database. Okay, so there we go. So they capture the information. This is the gross side of it, using the mouse anatomy ontology. So drop-down box, click on the tissue that's being collected, and then the gross anatomy is being categorized using the mouse phenotype ontology. So this is an appropriate level of capture for gross anatomy. And then they're qualifying that with terms from Pato. So now you know all about these ontologies, so this is all familiar to you. This is how important it is. So for example, you can put qualifiers in with the MP term, multifocal, unilateral, random, segmental, bilateral. And those are actually very important because you can actually use those to search your data with and to reason over your data. The histopathology inputs pretty much the same, except they use empath. So empath is the mouse pathology ontology. It could really be almost mammalian pathology ontology, which actually has all the appropriate terms when taken with the anatomy qualifier to describe pretty much any histopathological lesion that you can find at that level in the mouse. And so it's all being collected systematically. Similar systematic collection using these ontologies was done during a big aging program at the Jackson Laboratory, which is now just being analyzed. So 31 strains of mice were aged out completely with cross-sectional histopathology being done at 18 months and 24 months. So systematic pathology over all the strains. And the data all collected using empath and MA. And now it's being used to do GWAS analysis for predispositions between these strains to these different age-related diseases. So as soon as you have a way of coding, you can actually use that coding to use your data for computation. And if you were to make these diagnoses using standard narrative diagnoses, it would actually not be possible. So I think this, again, is the strength of using good, solid, reliable semantic methods of coding to code data even as complex as the pathology data. Okay, so I'm going to stop there and give you a list of the people who've been involved in the pathology side of things. The data I've shown you has actually come from John Sundberg and Peter Vogel predominantly. And leave it at that. Thank you.